Good morning, everybody. You can see from the little handout that I also spoke about newborn screening in San Antonio and Houston. So usually I say I get to talk about my two favorite subjects, genomes and babies, but today I'm going to try to just focus on genomes. So let's get started. Let's see. So these are our objectives for today. Hopefully at the end of this you'll be able to define the genome and the exome. You'll begin to talk about indications for when to do whole genome or whole exome sequencing, and then understand a little bit about the current challenges that we have when we look at a genome or an exome and don't quite know what to do with the results. So at first I'm going to set the stage. So why are we even talking about whole genomes and whole exomes? And then I'm going to talk a little bit about translational research. That's where we take the findings from the laboratory and begin to apply them to routine clinical care. Then they talk a little bit about best practices. So what are the leaders in the field doing today? What are they doing that they're not quite sure about? And what do we want them to do in the future? So what kinds of research projects should we be proposing to our academic partners and clinicians who do research? And then we'll spend a little bit of time on what we call the known unknown. So we know there are about 100 million variants across the 6 billion base pairs that we each carry. 3 billion from our mom, 3 billion from our dad. We don't know what those 100 million mean. So that's part of the known unknown. We really have to think about genetics in a new way where we're not quite sure what these variants mean and what does that mean for what you tell patients and their families. And then we're going to talk a little bit about some special considerations. So to set the stage, we're going to just remind you what human variation is, that the human genome is actually mapped, talk a little bit about the technology advancements that got us here, and then give you some definitions of genomic medicine. We talk about genomic medicine a lot. The sort of news anchors on CNN talk about genomic medicine a lot, so we want to give you a few definitions and really define the scope of what we mean when we talk about that. So just to begin at the basics, genetics is a study of single genes and their effects. That's what most of us learned in high school and undergraduate and our graduate or medical education. But genomics is really the study of all the functions of the genes and all the other parts of the genome. You know, we used to think that only 3% of the genome was coding and the rest was junk DNA. Now we know that that junk DNA is actually filled with molecular signatures that turn on and off genes at critical times in development and at critical times across our lifespan. Genomics not only takes into account the sort of linear base pairs of ACGT, but also what's going on in the environment. We're all born with a baseline genome, but then when you grow up in an inner city, you may have changes to your genome. If you work on a farm and are exposed to pesticides or insecticides, you might have a change to the genome. So we have to think about the genome as sort of a living thing across your lifespan. And when we think about genomic sequencing, when did you get your genome sequenced? Did you get it in a few hours after birth? Did you get it when you were 18? Did you get it when you were in the middle of your life? So we really have to think about genomic sequencing in the context of the lifespan. So just some basic concepts of inheritance after Dr. Rose's elegant talk. This is sort of like a really rudimentary slide, but just to remind us that 3 billion base pairs from our mom and our dad. As geneticists, we think about autosomal dominant, autosomal recessive, sex-linked disorders, all of the sort of genetic things that you think about. We think about these in the context of the genome as well. As we think about often when we find things we don't know, we begin to look at the parents and the extended family. So that's where pedigrees are sort of coming back into use in genomic medicine, not only looking at the patient or the proband, but also looking at the family and seeing what those tell us. As Dr. Friedenberg said, I was a member of the Human Genome Project, so my kids were little, and you know, I always had around the house the sort of traces because we were working 24/7 trying to beat every other lab to get our chromosomes done first. So I always focused on 4 letters, ACGT, and I had twins and they came home from preschool and they were so excited to tell me, Mom, there's more than just 4 letters. And they were so excited, and I said, I know, there's U, right? So ACGT, the T is replaced with a U when it's RNA. So, but I think they were talking about the whole alphabet. So